Greetings, I'm Gayatri from First MSc Microbiology, Sacred Heart College, Tirupattur. Today we are going to discuss about dengue virus. Introduction. Dengue is a mosquito transmitted virus and the leading cause of arthroborne viral disease in the world. It is also known as a breakbone fever due to the severity of muscle spasms and joint pains. Dandy fever or the 7 days fever because of usual duration of the symptoms. Dengue is found in a tropical and subtropical climates, mostly in urban and semi-urban areas. It is a problem in many tropical and subtropical parts of the world, including America, Africa, Asia, South America, and some parts of northern islands. In 1943, Ren Kimura and Susumu Hota first isolated the dengue virus while studying the blood sample of patients taken during the 1943 dengue epidemic in Nagasaki, Japan. Dengue virus is a single-stranded positive-sense RNA virus that belongs to the Flaviviridae family. It is an enveloped virus with 10,700 base pairs. Dengue is a member of the Flaviviridae family, which also includes other arthroborne viruses like Zika, West Nile, and yellow fever. Dengue is transmitted by Aedes mosquitoes. It characterized as a arbovirus, arthropod-borne virus, because it is transmitted by mosquitoes. Structure of dengue virus. Dengue virus is 40 to 60 nanometer in size, which is isometric nucleic acid of 25 to 30 nanometers. This dengue virus structure is roughly spherical with a diameter of approximately 50 nanometers. It consists of a capsid, envelope, membrane, and genomic RNA with 100 angstroms. It is an enveloped virus. Types of dengue virus. The four dengue virus serotypes are dengue1, dengue2, dengue3, and dengue4. They are called serotypes because each has different interactions with the antibodies in human blood serum. In 2013, a fifth variant, dengue5, was isolated. This serotype follows the sylvatic cycle, unlikely the other four serotypes which follows the human cycle. The replication cycle of dengue virus. The cell receptor binding. The cell enters into a host cell and endocytosis process is occurs. Fusion of the cells then moves to the nucleic acid capsid release. Then RNA replication and transmission of capsid and the spike proteins. Then binding the endoplasmic reticulum to the Golgi apparatus. The fluorine cleavage was occurs. Then the release of progeny virus particles. The virus receptor, mature particles and immature particles are occurs. Then the release of progeny virus particle leads to the infection. The replication cycle of dengue virus includes the following steps. Virus binds to the host cell receptor, enters to the host cell via receptor mediated endocytosis. Virus is neutralized into a clathrin-coated endosome. Viral genome is transported to the endoplasmic reticulum, replicated in the viral proteins are synthesized. They are assembled into a nucleic acid, the mature and aggressors. Then newly synthesized dengue viruses are released and go to the infect other host cells. Pathogenesis of dengue virus. First, infection in dendritic cells. Due to the enters of dendritic cells in the skin via mosquito bites, the dengue virus can directly include the dendritic cell apoptosis through the replication inside the infected cells. Disseminated via lymphatics. The virus disseminated via lymphatics to the other tissues. Other mosquito bites cells in the skin and infectedly delivered dengue virus and draining the lymph nodes. The lymph nodes contains the resistant macrophages and other unknown cells. These cells infect and delivers the virus to the lymphatic and vascular system, which can lead to the infection on bone marrow. Transmission of dengue virus. A mosquito bites. The dengue virus is spread through the bite of an infected mosquito, primarily the Aedes aegypti mosquito. Other species within the Aedes genus can also act as a vectors, but their contribution is secondary to Aedes aegypti. Blood transmission. The virus can be transmitted through blood transfusion or organ transplantation. Clinical symptoms of dengue virus. Fever. A high fever at 104 degree Fahrenheit and 40 degrees Celsius. Headache, a severe headache, pain, a behind the eyes, muscles, bones, and joint pain, or extreme itching in the legs and joints. Then rashes, a rashes that appears 2 to 5 days after the onset of the fever. Other symptoms like nausea, vomiting, fatigue, swollen glands, and leukopenia, and a positive tourniquet test. Laboratory diagnosis of dengue virus. Serological test ELISA, molecular tests like PCR, RT-PCR, complete blood count, erythrolytic test including potassium, sodium, chlorine, liver function test including ASLT, ALT, GGT, albumin test. Treatment: there is no specific medicine to treat dengue virus. Prevention of dengue virus: eliminate mosquito breeding site, use mosquito repellents, wear protective clothing, mosquito nets, stay indoors during the peak mosquito activity. Thank you. Thank you for your patience listening. Thank you.